today's lecture, as I said, is going to be about nucleotides. We're going to have a couple slides about some derivatives, let's say folates, biosynthesis, for folate biosynthesis or NAD, but the bulk is nucleotides. It's an extremely important part of metabolism. It's very close to home for me right now. The reason is because every proliferating cell has to make a lot of these things to incorporate them into its nucleic acids, RNA, DNA especially, and pretty much any intervention that interferes with these enzymes will delay or stop DNA replication, which means it will delay or stop cell proliferation, which means it will delay or stop inappropriately proliferating cells, such as cancer cells, for example. And those are chemotherapeutics. So these compounds are of extreme public health relevance, and these pathways, as I said, are super important. But we'll go through this. So what is it that we're going to cover? We're going to discuss how you make pyrimidines and purines. Of course, these two are the two general classes of nucleotides, depending on the base that they contain, the nitrogen base. The smaller ones are pyrimidines and the bigger ones are purines. Now, cells not only make from scratch these compounds, and all cells have to do this, as you'll see. They have to be able to make it. So this is not like amino acids where we only make 10 of them, half of them, and the rest we have to get them from our diet. Here, every cell, including human cells, they have to make their own nucleotides. But given the option, they can salvage bits and pieces that they can convert into the nucleotides they need. So they can convert and modify them in a way that they can be used to make the purines and pyrimidines they need. Then we're going to say a few words about the breakdown of these compounds, about these nucleotides, and because again, that's of public health relevance, as you will see. And as I mentioned earlier, we're going to close with some cofactors that are related. So what are these pyrimidines and purines? How do they look like? I'm sure you've seen them before. The pyrimidines are the small ones, one ring. Okay, and purines have two rings, and you can see all the nitrogen. So these are very rich in nitrogen. The pyrimidines that we're going to be talking about, there is cytosines and uracils. Cytosine and uracil are found in nucleotides that are building blocks of RNA, and instead of U, we have T's in DNA. Okay, that's the difference. The only difference between U and T is really this methyl group here. Purines come again in a lot of flavors. These are the ones that we're going to be talking about that make it into nucleic acids, A's and G's. But there is also hypoxanthine and xanthine that are important intermediates, both in the synthesis and breakdown. And uric acid, super important. This is what we excrete, in our urine. But there are other ones that we take in our diet, such as caffeine, for example. Okay, it's a very interesting compound with amazing properties. Something related also in tea and coffee, also theobromine. Just to remind everyone what we're talking about. All these things that we're talking about, the purines and pyrimidines, they are this base, the blue thing that's attached through a glycosidic bond to a pentose ring. And in the 2-hydroxyl group is a hydroxyl in the ribonucleotides, so the ones that make into RNA, let's say. But in DNA, this is reduced. Okay. So that's the nucleoside. Okay, if you have a base and the sugar, you are dealing with a nucleoside, and again, the base could be any one of these things. And then depending on how many phosphates you have 1, 2, or 3. Those are all nucleotides, and whether it's a monophosphate, a diphosphate, or triphosphate, that makes it. If it's an A here, it makes an ATP, ADP, or AMP. Okay. And we've talked previously. How let's say UTPs used to activate sugars, you know, CTP how to activate lipids, and so on and so forth. So, and adenylation and uridylation, you know, all over the place to modify proteins and so on and so forth. As I said in the beginning, all cells have to make nucleotides from scratch. Why is that? Well, I mean, the negative charge, why can't you just eat ATP and be done with it, you know, to cover your energy needs and eat all the others too to make your DNA or RNA? Well, the negative charge makes them impermeable through the plasma membrane. So no matter what you do, you won't be able to absorb them.